Cell growth and division. Limits to cell growth. There are two main reasons cells divide instead of continuing to grow larger. The larger the cell grows, the more demands the cell places on its DNA. The cell will also have more trouble moving nutrients and waste across the cell membrane. Division of the cell. Before a cell becomes too large, it will divide, forming two new daughter cells. This process is known as cell division. Before the cell divides, the cell copies its DNA. Each daughter cell gets one complete set of DNA or genetic information. In prokaryotes, cell division is simply separating the contents of the cell into two parts. In eukaryotes, cell division is more complicated and occurs in two main stages, mitosis and cytokinesis. Cells produced by mitosis are identical to the parent cell, therefore it is classified as asexual reproduction. Mitosis is the source of new cells when multicellular organisms grow and develop. Chromosomes carry genetic information from generation to generation. They are made up of DNA and proteins. They are visible during cell division because they condense into compact structures. Normally they are not condensed and they are spread throughout the nucleus. Before cell division, each chromosome is replicated. Because of the division, each chromosome consists of two identical sister chromatids. The sister chromatids separate from one another and one chromatid goes to each of the two new cells. The pair of chromatids are attached at an area called the centromere. Centromeres are usually located near the middle of the chromatids. This is a picture of a chromosome. These are your sister chromatids. Here is your centromere, and kinetochore is a protein that is located on the centromere. I would like you to answer these 5 questions and turn them in with your notes. The cell cycle. A series of events that cells go through as they grow and divide. During the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells, which begins the cycle again. The cell cycle consists of 4 phases: G1 phase, S phase, G2 phase, and M phase. This is a picture of the cell cycle. It includes interphase as well as cell division or mitosis. Interphase is a long process that includes G1, S, and G2 phases. During G1, cells do most of their growing. They increase in size and synthesize new proteins and organelles. During the S phase, chromosomes are replicated. Synthesis of DNA molecules and key proteins associated with chromosomes are also made. During G2 phase, organelles and molecules required for cell division are produced. Mitosis is normally divided into 4 phases, PMAT. Prophase being the longest phase. Chromatin condenses into chromosomes, centrioles move to opposite sides of the nucleus, and spindle fibers begin to form. The nuclear envelope also breaks down. Prometaphase is a phase where the nuclear membrane dissolves, marking the beginning of prometaphase. Proteins attach to the centromeres, creating the kinetochores, and microtubules attached at the kinetochore, and the chromosomes begin moving. During metaphase, chromosomes line up across the center of the cell. Each chromosome is connected to a spindle fiber at its centromere, or kinetochore. Anaphase, the sister chromatids separate into individual chromosomes and are moved apart. During telophase, the chromosomes gather at opposite ends of the cell and lose their distinct shapes. 2 new nuclear envelopes will form. These are the phases of mitosis. Prophase, you can see that the nuclear envelope is beginning to break down. chromosomes are visible, so that means that that DNA has condensed and has coiled around those associated proteins. These centrioles are beginning to move to opposite ends of the cell. Here we see metaphase. One of the key things you want to remember about metaphase is that the chromosomes line up in the middle. These blue lines represent spindle fibers. Anaphase, we can see that the chromatids are being pulled to opposite ends of the cell, and with telophase, we can see that nuclear envelopes are beginning to form, and you will eventually have two individual identical cells. Cytokinesis occurs at the same time as telophase. In animal cells, a cleavage furrow will form. The cytoplasm pinches in half. Each new daughter cell has an identical set of duplicate chromosomes. So in animal cells, cytokinesis involves a cleavage furrow, and in plant cells, you will see a cell plate. In plant cells, a cell plate forms midway between the divided nuclei, gradually developing into a separating membrane. A cell wall then begins to appear in the cell plate. So when you think of cytokinesis, cleavage furrow takes place in animal cells, and cell plate takes place in plant cells. Alright, here's another set of questions, so that will be a total of 10 questions that I would like you to answer and turn in with your notes. If you have any questions, please contact me through Remind or send me an email, and have a good one.